Simon Lovell and Simon Wheeler, and we published it last year in Molecular Biology and Evolution. And recently I was appointed a lecturer in the Faculty of Medicine and Human Studies, so in the Institute of Cardiovascular Science.
So how do you guys, British people feel about the Brexit? Because it's never come, changing a little bit. People, some other people may be interested in this. Yeah. It's a hard topic to discuss.
There are some concerns at the university level, but there is nothing clear at the moment. So it's going to affect funding or academics working in the UK. So there's nothing else.
So it's a tough time for you guys to do research in data. So did you have a good time here?
Yeah.
Enjoy Disneyland? Disney World. Usually come here for a while and start on that time. So I think now it's a good time to start.
Okay. So hello, good afternoon. As I said, I'm going to talk about this paper that we published in Molecular Biology and Evolution last year. And let me start with a couple of definitions, because covariation and coevolution have been mixed up in the past. Covariation is the observation that particular pairs of amino acids co-occur more frequently than expected when we look at two columns in a multiple sequence alignment. And coevolution is an evolutionary process whereby amino acid substitutions at one position within a protein affect the selective pressure acting on nearby positions, and hence affect their substitution rate. And it's widely assumed that coevolution causes covariation patterns, and although the exact mechanisms are not well understood, is believed to be because of an enrichment in correlated mutations that improve the fitness of the organism. And it's also believed that covariation can be used in order to predict coevolution. So this has encouraged the development of a wide variety of covariation metrics and tools that attempt to identify covariant positions within a multiple sequence alignment in order to infer co-evolving residues within a protein. And all this interest in coevolution and covariation is raised by the fact that co-evolving residues are expected to be in close proximity within the protein structure and to be functionally related. As a consequence, the identification of co-evolving residues is very interesting in many areas of research. For example, as a way of generating constraints for a finished polygon of proteins in protein design, and in attempts of predicting protein interactions from protein sequences. There are mainly two coevolutionary scenarios that we can think of. In episodic coevolution, the first mutation causes a decrease in fitness that is recovered by a second compensatory mutation. This process is time reversible. This means that it can progress in both directions. And this results in an ongoing cycling between optimal and suboptimal states. We expect that this is going to generate lots of double changes within a phylogenetic tree, and also that it's going to produce a strong covariation signal. In the case of directional coevolution, the first mutation doesn't have a great fitness effect. However, it changes the structural conditions in a way that it allows a second mutation to occur. This second mutation, which improves the fitness of the organism, may even be detrimental if that first mutation hadn't occurred before. This type of a scenario has been identified in cases of antibiotic resistance. And it can only progress in that direction, that is, the improvement of the fitness of the organism. For these reasons, it's not expected to occur very often in an almost constant environment. So we believe that the contribution of directional coevolution to the covariation signal is going to be restricted to a few specific examples. So we were interested in the evolutionary basis of covariation, and we were concerned about how three independent metrics could differentiate between different evolutionary scenarios. For example, these two examples. So these are examples of a pair of binary sites. And the distribution of observed states in the leaves of the trees are the same. And hence, the covariation measured here as model information is equal. However, the succession of evolutionary events leading to those observed states is different in both the scenarios. In scenario A, after that initial divergence, there is complete conservation. This means that the signal generated in that initial split is amplified by the tree structure. In scenario B, there are multiple instances of double changes represented by this red full disk. So this will be a good example of the episodic coevolution that I mentioned before. And as single changes occur, here single changes are represented by this red half disk, the covariation signal starts to fade and can even disappear. However, again, these pairs of scenarios are quite different. So on the scenarios on the left, after that initial divergence, there are only single changes. So this suggests that those sites are evolving independently. On the other hand, on the scenarios on the right, there are still more double changes than single changes. And this supports the idea that this might be coevolutionary scenarios. So in our examples, the increase in the number of single changes causes a decrease in covariation. And this is independent of the number of double changes that have occurred. So this demonstrates how sensitive is covariation to the number of changes and the position within the phylogenetic tree where they occur. So this makes us conclude that probably covariation is not appropriate for us studying coevolution. Anyway, we were interested in investigating which of those scenarios were more likely to be found. So we set up a model of binary coevolution. So we have, again, two sites, and each site can have two states, 0 and 1. So there are two optimal combinations of states, 0, 0, and 1, 1, and two suboptimal combinations, 0, 1, and 1, 0. In order to progress from one optimal combination to the other, two substitutions are required, but we don't allow these substitutions to occur simultaneously. So this means that in order to have a double change, one substitution has to go after the other. This makes that the amount of time that two sequences are allowed to diverge is very important for their probability to accumulate substitutions. This scenario can be modeled as a Markov process, and if this was an independent process, the instantaneous substitution rate would only depend on the frequency of the single states 0 and 1. But if there is coevolution, the difference in fitness between the optimal and suboptimal states is the strength of coevolution, and this depends on the effective population size and the selective pressure. In that case, the instantaneous substitution rate depends both on the frequencies of states and the probability of fixation of a new mutation. So we investigated the behavior of that model using different combinations of parameters. So in this plot, the x-axis shows the strength of coevolution. 0 means that it's an independent process, and coevolution increases in that way. This gray line here shows the relative evolutionary rate. That is, the evolutionary rate when there is coevolution compared to the rate of the independent process. And what we see is that when coevolution increases, the evolutionary rate rapidly decreases. these black dashed lines represent the proportion of single changes observed under different divergent times. So for similar sequences, the vast majority of changes are single changes. For distant sequences, here I'm referring to time equals 1, we need a strong coevolution in order to get 50% of double changes. And in order to achieve that expected enrichment, clear enrichment in double changes, we will need very strong coevolutionary forces and unfeasible evolutionary time frames. So this model predicts that we are not going to find many double changes in the phylogenetic tree, and that coevolving residues are probably evolving at a low rate. The problem here then is how we can differentiate these from two conserved sites evolving independently. So then we decided to test some of our predictions analyzing covariant positions within well-defined phylogenetic scenarios and big alignment datasets. So we use several covariation metrics in order to select different subsets of covariant positions. We found that in all cases, most of the covariation signal was generated by independent processes. So these single changes that occur in different branches of the tree. And probably, this suggests that probably this signal gets amplified by the tree structure. And in order for this amplification to occur, these sites should be quite conserved. So we found that most of the methods, especially the best performing ones, select sites that are evolving slowly. The only exceptions were mutual information and chi-square, which select pairs of fast evolving sites. But this is interesting because it can be used in order to explain the different performance between covariation metrics. Because it's well known that the most conserved residues in a protein are packed in the core of the structure, whereas the residues in the surface are less conserved. And this is exactly what we found. So mutual information and chi-square select pairs of surface residues, whereas the best performing methods select residues that are in the core. So our findings have implications for the use of covariation in protein folding and function prediction. So we have found that covariation methods select pairs of varied residues with low evolutionary rate. This is clearly not the way that they are intended to work. However, having said this, if you are interested in finding residues that are in close proximity in the structure, some of these methods can do this very well. Just be aware of their biases. This low evolutionary rate also explains why these methods need big alignments. And the issue of getting enough variation is also relevant if we are interested in studying particular sites that may be very conserved. For example, catalytic sites. In those cases, even in massive alignments, there are going to be very few changes. We believe that this difficulty might be overcome by combining covariation methods and phylogenetic approaches such as maximum parsimony in order to include these extremely conserved sites in the analysis. And knowing that surface residues are less conserved than core residues, we believe that all the current methods are going to struggle for predicting interaction interfaces unless we can use prior structural knowledge in order to filter the core residues out. And anyway, they are going to do better at predicting obligate complexes than transient interactions. Our findings also have implications for the study of molecular coevolution. The most obvious one is that covariation methods shouldn't be used for studying molecular convolution because they cannot differentiate between evolutionary scenarios. But the most interesting one is derived from that drop in evolutionary rate. So we believe that this suggests that the stronger the coevolutionary selection, the shorter the time for the compensatory change to occur before the first mutation gets purged from the population. And this leads to the coevolution paradox. That is, the stronger the coevolution, the less likely that we are going to observe it. Just before finishing, let me thank Simon Lovell and Simon Whelan for their support, supervision, and hard work, and BBSLC for funding. And as I said, I recently got appointed a lecturer in the Institute of Cardiovascular Science. If you have any comments, suggestions, or shared interests, just don't hesitate to contact me.